This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 529, recorded on December 11th, 2018. I'm Vincent and you are listening to the podcast all about viruses. Today we are recording in Madison, Wisconsin. I'm at the University of Wisconsin, my fifth or sixth or seventh visit here. Come back here a lot, not for the weather, but because there are a lot of good scientists here, and I enjoy speaking with them. And today I have a very, very special guest, someone I've been wanting to have on TWiV for a long time. He's an emeritus professor from the University of Wisconsin-Madison, Roland Rueckert. Welcome to TWiV.
Thank you, Vincent.
You know, Roland, you had a big impact on my career, especially early on. And so I always wanted to have a little conversation with you on the show. And I remember very fondly, we were at a meeting in Switzerland, Arola, up in the mountains. And you and I took a long hike together. I could barely breathe because it was way up in the mountains. And we were talking the whole way, and then we sat in the back of the hotel for a couple of hours until the sunset. And that was really great. You probably don't remember that, but I do.
Actually, that sounds wonderful. I'm sorry to say I don't remember that.
It's okay. But I'm sure you'll remember lots of other things. And so I wanted to talk a little bit about your training and some of your science today. And let's start all the way at the beginning. Tell us where you're from.
Well, I'm pure Wisconsin. I was born in Rhinelander, Wisconsin, which is 200 miles north of here. Then I came to the university in 1949, 4 years after the end of the Second World War. The place was a broil with veterans who had families and were all trying to make a living and start new lives. It was a wonderful time, and I still remember with great gratitude those soldiers who came back. Then I was interested in chemistry. In fact, I was a little bit manic about chemistry. I sold seeds for the Park Chemical Company, and I got a chemistry set for a prize, and that set my whole career. And if you ask me why, I have no idea. I just liked chemistry.
You told me you lived in tents on the campus, right?
Well, the campus was so crowded with veterans that there was no room in the dorms. So they literally had tent colonies, and they had places way out at the Badger Munitions place out by Baraboo. So the place was really jammed with veterans, and we had huts that they call Quonset huts. We had our classes in there. So it was a completely different place than it is now.
So we're on the top of the Bach building here, and you showed me earlier, you eventually moved to a building near the lake, and you could walk up the hill every day thinking about chemistry, is what you told me.
I was absolved from the tent colony at the last second. They put me in Trip Hall, which is down on the lake, right down at the bottom of the hill. And the chemistry building was over on the other side of the hill. And every day I would walk up over the hill. I was young then, and walking up the hill was easy. And my heart was so singing because I was going to study chemistry, learning about chemistry. I have no idea why. It's just what I wanted to do. Well, I do know. My dad had a grocery store, and it was open 7 days a week. And when I was studying chemistry, I was not working in the grocery store.
That's great. So you graduated what year?
I graduated in 1953 and was immediately conscripted by the Army. So I remember weeping as I left and looked behind. I was at the McArdle, I had a job at the McArdle laboratories, thinking what a disaster it was. I went into the military, the artillery. I can't remember, Fort Riley, Kansas. Did basic training and hated every minute of it. Then they sent me to Camp Pickett to be a medical aid man. And that turned out to be good, actually. I learned a lot of things about medicine there that stood me in good stead when I came back here. So I changed my mind about the military. I came to see it was necessary, and there were honorable people there. But I lost 2 years. Actually, it turned out they made up for it. When I retired, I got those 2 years towards my pension, so I didn't lose any time at all. So partly, Congress was generous then. I don't think they're that generous anymore with the military.
So in 1953, you graduated. That's a big year in science, you know.
Well, remind me.
Structure of DNA.
Oh yeah, 1954.
And there's something else that I've forgotten in 53. And of course, I was born in 1953.
Ah, now I got it.
There you go.
There are other things. I'm collecting a list of science milestones for 1953, and I don't remember the others, but DNA is obviously a big one. And you graduating from college, that's big too.
Right.
So 2 years in the Army, then what did you do?
Well, I came back to McArdle. They wanted me to come back as a graduate student, so life was pretty easy. I didn't really have to go applying for anything. I got a good graduate student thing, and we did research. I worked for a professor named Gerald Mueller, who had his fingers in many pies. So I learned about a lot of things, estrogen receptors, but he was interested in technology. Gel filtration was new in those days, ion exchange. I actually began working on tissue culture. As a matter of fact, I was the first person at McArdle to work on tissue culture. The first person was Gilbert Chang at the State Lab of Hygiene. He was growing poliovirus, and I got my cells from him. And we worked on learning how to grow HeLa cells, which was pretty miraculous. And at one point, I tried cultivating cells from primary cultures and took mouse embryos and took brain and kidney and heart and liver and tried trypsinizing collagenase, chymotrypsin and not chymotrypsin, trypsin, yeah, chymotrypsin. And I got all kinds of cells under the microscope. I remember vividly liver cells that had saddle-shaped cells under the microscope. And I tried forever to get those again, and I was never able to reproduce that again. But it was a new world. I mean, here we had tissues, and we could actually grow them in culture like microbes, and I understood this is the beginning of a revolution. But I couldn't think what to do with them. The cells didn't grow very well. I had heart cells that were actually beating like that. It was really an exciting time, but I didn't know how to exploit it. But I did go on with the HeLa cells, try to do genetics with them, and I studied something called thymineless death, where if you deprive a cell from its ability to make DNA, it will die. When it makes the DNA, if it cannot make protein, it will live. But if it makes protein, it'll die. So that was the basis for a selection method, but I didn't have much to select. So I understood I could do some genetics. I published a paper in Cancer Research. And unbeknownst to me, it became a citation blockbuster. I didn't know anything about it because I'd gone to Germany as a postdoc to study protein synthesis in cell-free extracts. And first I started with flu, and I didn't want to work with flu because it was sort of an ill-defined thing. It didn't have a nice sedimentation constant and so on. So I switched to another virus called ME virus, which is a single-stranded polio-like virus. And I could get that out, and I could show that it was infectious, which was key, because if I was going to prime protein synthesis, I wanted to know that my messenger was intact. So by using ME virus, I hope to build a protein synthesizing system. And I had one trouble after the other. I got infections in my cells, and they wouldn't make plaques sometimes, and I had a lot of technical difficulties in Germany, but gradually I made progress, but never with the cell-free extracts. Never was able to make any headway with that. And then one day, now eat your hearts out, I get a letter from Berkeley, California, from Wendell Stanley, Nobel Prize winner for crystallizing tobacco mosaic virus, head of the virus laboratory. Would you like a job at the virus laboratory as an assistant professor? I'd never taught a day in my life, and I'd never had a virology course, and he's inviting me to be a assistant professor in virology. And I thought, I can't do that. So I told him I wasn't prepared to be an assistant professor. He said, well, how would you like to be a virus assistant virologist? That was fine. You got a job. So without even applying, I got a job at the virus laboratory, sort of science fiction.
What year was that?
That was 1962.
So who in Germany did you do a postdoc with?
With Werner Schaefer.
Okay, okay.
You know Werner Schaefer?
I don't know him, but I know the name, yeah. I found what I think is your first publication, 1956, The Effect of Hypothermia on Morphine Metabolism in an Isolated Perfused Liver.
Well, okay, that was in the Army. I skipped all that.
Oh, but it's okay. That's the first paper that shows up on PubMed.
Yeah.
Then there are more papers in between, and then in 1962, you're a first author on a paper, An Improved Procedure for Purification of Bacteriophage FIX174.
Right. I went to Munich first to work on bacteriophage because I had no experience at all with viruses, and I wanted to work with animal viruses, but I didn't know anything about virology. So I thought FIX174 would be a good way to get an education.
And you did that before you went to Berkeley?
I did that before I went to Tubingen, which was Werner Schaefer's laboratory.
Okay.
So I went from bacteriophage to influenza to ME virus to Berkeley.
You speak German?
Jawohl. Ich könnte Vortrag geben.
Okay.
I said I could give a lecture.
In German?
Auf Deutsch, yeah.
Wow, that's great. All right, so then you go to Berkeley. What year was that now?
That was 1962.
What did you do there?
Okay, I kept working with ME virus. And I was purifying it, the same project, trying to get enough RNA, using a phenol procedure, which is a new method. And it happened that Frankel Conrad was there, and he had made infectious RNA from TMV.
Right.
And who was it now? Gierer had made infectious RNA by the phenol method. You shake with phenol, you get two phases. Protein goes into the phenol phase, the RNA goes into the aqueous phase, and the phenol gets rid of the ribonuclease, so you can make infectious RNA that way. And Frankel Conrad was making it with SDS. He said he was fuming and sputtering, and he said, you can't make biologically active materials with phenol. Phenol is a poison. It kills bacteria. So there was this fight going on, and Frankel Conrad was there, and I had just come from Tubingen, where his competitor was working. So he was very interested in talking to me. And he adopted me. And I needed to be adopted because I was all alone, and I needed somebody to talk to about virology. The other virologists were Gunther Stent and some other high-powered guys that were phagemen, and they really weren't interested in animal viruses because that was pretty passé. So I had Frankel Conrad for a colleague, and he helped me along. So I was able to do some protein chemistry through his help. He had an amino acid analyzer, and so I was able to publish a paper on the proteins of ME virus and show that there was more than one and things like that. So it was pretty primitive chemistry, but that was 1962.
Yeah, so I have a paper here, 1965, on the proteins of ME virus. It's you and Walter Schaefer. It's a virology paper.
Yes, so Schaefer gave me the virus and gave me the project, though. I owed him that loyalty.
So there are some other early papers, too. Some of the co-authors include Peter Duesberg.
Yes, Peter, believe it or not, was my assistant at Berkeley. So Stanley came to see me, and he wondered if I could accommodate Peter Duesberg in my laboratory. So when Peter came, he found out about this phenol technique. And I had Harry Rubin working right next door, and I told Peter that I thought that it would be interesting to try the phenol technique on this Rouse sarcoma virus and see if we couldn't get infectious RNA out of the Rouse sarcoma virus. But first I wanted to finish this project on the ME virus protein. So Peter, without consulting me, just went and got my virus and my Rouse sarcoma virus and phenylated it and proceeded with the sequencing. So he was very aggressive. There was nothing I could do about it. I mean, I was at fault because I was holding back and finishing a project before doing this other project. So he essentially took it over by being aggressive. I had my own project. I could live with that. So he came out and he found this sequence and he said, here's an oncogene. As far as I know, that was the first person that claimed that he had an oncogene. And the concept that one protein could cause cancer was pretty radical. But then that project got taken away from the guys in San Francisco. That was Mike Bishop and Harold Varmus. They basically took over that project. And Peter went off on his own way. He, for some reason, denied that that the virus had anything to do with HIV. I never understood his thinking. I think he just liked to be a radical.
Yeah.
But he had trouble. He ran into trouble.
Yeah, I noticed some of the early papers are on retroviruses too, besides the ME virus.
Well, I tried actually to get involved with tumor viruses, but I found that the research competition was too much. When I applied to work on retroviruses, they said, you're a picornavirologist. Why are you working on retroviruses? Well, obviously the answer was because I wanted to get into it. But that wasn't acceptable. So I had trouble maintaining a presence in retrovirology.
Okay. So how long did you spend at Berkeley?
3 years. And then I got another letter from Paul Kaysberg, would you like a job at Wisconsin? So wouldn't you like that? I mean, the world is different now.
Sure is. You have to really work hard to get jobs.
Yeah. So in fact, I was the first one to move into this building. It was finished in 1965. And I actually, you see those columns there? They're made out of special hard steel reinforced concrete, and I needed to put a coat hanger up so that when I came to work, I could hang up my coat. That's how new the building was. And I spent one drill for each hole so that I could get the roll plugs in so that I could put my coat rack up there.
So what year was that you came here?
That was 1965 in July.
So you brought viruses with you and you continued picornavirus?
I continued with the ME virus at that time, yes. And then I started working with the MC virus, which I liked better because it made much better plaques. It was a more aggressive virus, and easier to work with.
So I found a 1969 paper, The Structure of ME Virus, a Model. It's you and C. Stoltzfus. That's from here, right?
And Keith Dunker. Keith Dunker should have been the first
He's there, yep.
author on that paper.
So what's that about? At this point, you're interested in understanding how viruses are built?
Well, okay. So you remember the project was to study protein synthesis and
Right.
cell-free extracts. And the idea was we were going to make an antibody against the coat protein, which, by the way, was a bad idea. That would not have worked. But I made antibodies against the virus, and I wanted to know how to recognize the protein when I had it. So I needed some method, and I decided electrophoresis. That was the reason for characterizing the protein. So then we developed electrophoretic methods for doing that. So we were able to dissociate the virus in a quite miraculous way by treating it in a special condition we could generate 14 S subunits, and with treating that with urea, we could get 6 S subunits. And by characterizing them, because we could show that the poliovirus was built of 60 subunits that were put together in pentamers. And so that's what we published. And although we thought that was pretty marvelous, it settled the question, because the electron microscopy meant you could show that there were 60 subunits, you could show that there were 120 subunits, and you could show that there were 90 subunits. And we found that because of the tripartite nature of the virus, that you could explain all three structures depending on how the proteins segregated when they dried. So that was the subject of that particular paper.
So you mentioned using gel electrophoresis. That was one of the important methods of the
So
time,
while
right?
I was at Berkeley, the idea was to get the protein, get the RNA out intact, and since the protein went into the phenol, we could recover it from phenol and then try to characterize the protein. So the trouble was the protein was completely insoluble, and that was basically the same story for every virus that was known. They were all insoluble, all the coat proteins. So we have to dissolve them in something, and 8 molar urea was the protein solvent in those days. So we put that into the electrophoresis unit, and we could separate subunits that way. We worked with that, and then Jake Maisel published a paper with SDS, and he said, "Oh, there's 3 peaks in here in SDS," and I thought, that can't work. There's SDS is loaded with detergent, and you can't separate anything with in SDS. So I talked to my students about that, and we decided we're going to prove that you cannot do the molecular weight in SDS. And the way we're going to do this is I don't know, this is kind of intricate. Lysozyme, I had a student lab experiment where we dissociated lysozyme, which has 4 disulfide bonds. And by substituting these disulfide bonds with disulfide mixing, we could put in cysteine, which has plus-minus charge. We could put in cysteamine, which has just a plus charge on it, or we could put on cysteic acid, which has a negative charge on it. So we could put 16 negative charges on, 16 positive charges on, or 16 zero charges on. And we put all these in the SDS electrophoresis to show how they were not going to separate properly. And lo and behold, the charges were irrelevant, and all of the lysozymes migrated at the same rate. So indeed, it did it according to molecular weight. So we then finally realized this had to do with the relationship of the number of duodecyl groups that were bonding. And we did this with a whole bunch of other proteins, and we published it in JBC. And as it happened, there was another fellow that was 2 months ahead of us who published the same result. I forget his name. Do you remember his name?
Was that Lemley?
No. But anyway,
Nobody remembers.
we were scooped, but it turned out to be a citation blockbuster paper. Anyway, we got 1,000 reprint requests.
Reprint request, what's that?
Well, that's the way we did business before computers.
So you published a paper in 1972 I wanted you to comment on. This is a great title. An automatic high precision acrylamide gel fractionator. What's that?
Well, okay, we had these gels, and we could separate all of the proteins in them with the radioactive peaks that were in there. But getting those gels we wanted to be able to count them. And this was before putting things on photographic plates. So what we did was we pushed the gels out and cut up the segments, and then we measured the radioactivity in each one and then made a plot, and we could see the proteins. And this gave pretty rough gels and took a lot of time. So I went to see Gilson Medical Electronics, and I told him that we could measure the molecular weights of proteins by electrophoresis in one day. It took a thesis, PhD thesis, to do one protein, to do lysozyme. That took 4 years. So he was pretty astounded by that. And I told him we needed an automatic machine that would, do this more quickly. So Warren came over and he looked at the setup and he said, "Oh, I can build you a machine for that." He basically made a plunger and put a little hamburger grinder down at the bottom, run it through a wire, and then collected it in a fraction collector of his making. And it worked like a wonder from the very first day. So he made one model, and it worked like a wonder the first day. So I said, "You can have the patent. We just want the machine." And he gave us the machine and was happy to have the patent. I don't know. He may have done well on that.
So just to make it clear, these are gels you ran in tubes.
These were gels we ran in long tubes, and there was a bit of a science to cleaning those tubes properly so the gels didn't crack when you made them. And actually my students worked that out, and I don't even know how they did it, but they managed to make those gels so they wouldn't crack when you made the long tube.
You had the long tube, and then you would push the gel out.
We'd push them out and
slice them up, right?
And that sliced them up, and we'd collect the fragments in tubes according to their distance.
I saw once a machine which was basically a series of razor blades, which you would put down on the tube and slice it up, and then you take each one and put it in.
Right. So that was the way it was done before the machine.
Right.
And after the machine, people at meetings would come up in awe and say, "Oh, that's a wonderful machine." And that was the technology for about 1 year. And then putting things onto photographic gels came, and that was obviously superior. That was a much better way to do things.
And to do that, we had to have flat gels then, right?
We needed
so
flat
we had,
gels.
and that's what we run to this day, flat protein gels, which you can then put on a. And we don't use film anymore. We have non-radioactive detection methods.
Oh, well, okay, I didn't know that.
That's right. There's very little radioactivity used anymore. It's just amazing. Okay, so moving to 1973, and this is, I think, a really important paper, and I want to know how you got interested in this. It's Journal of Virology, Picornaviral Gene Order, Comparison of a Rhinovirus with a Cardiovirus. So how did you get interested in gene order?
Well, let's see. You really have the dates there. That's great. At some point, somebody asked me to be chair of the Picornavirus classification system, the International Congress. And so I'm looking at all these things, and we were beginning to do rhinoviruses at that time, and the rhinoviruses were classified as picornaviruses, but so was notomorvirus, and that seemed to be different. And so the question was, are rhinoviruses really picornaviruses? And so we became interested in how do you compare viruses to see how they're related, and we thought gene order should be a pretty good method of comparing things. That's how we got started in it, was to compare different viruses. We had ME virus, and we had EMC virus, and we had, I forget, a number of other viruses in the laboratory. All the rhinoviruses. We had a number of rhinoviruses. And we were able to grow all these things and radio label them, so it was pretty straightforward to do the comparisons that way. And it was pretty clear immediately that they're all picornaviruses. And that left notaviruses. What about them?
So you at this time, do you already know that these proteins, these viral proteins, are made as one long precursor?
Yes, so David Baltimore had shown that, and it was pretty easy to reproduce his experiments. So that's basically, we just followed David's work.
So then there are a series of papers about processing, and there's one in 1979, and the authors are Ann Palmenberg, who's here, and Mark Palanch, and it's protease required for processing the coat protein resides in the replicase gene. So at some point you got interested in proteases.
Well, they were getting all cleaved up, so the question was, who's doing the cleaving here? Mm-hmm. Is it a host protein or was it a virus protein? So that was the basic question. And I can't remember. I do remember we had discussions in lab meeting about how to test that, and we decided that dilution was the way to go to see if it was a first order or a zero order reaction. And Ann did that experiment. And I think immediately, as soon as she did the experiment, it was clear it was zero order. It didn't matter what the dilution was. They cleaved at the same rate. So it had to be a virus-coated protein.
So was Ann a student at that time in the lab?
Were you a postdoc, or you were
Postdoc.
You were a postdoc?
Okay. And Mark Palanch is on that paper, and he's now at CDC.
Yeah, I can't remember what Mark's involvement was. Do you remember?
He taught me how to run gels.
Oh,
Okay. He taught Ann how to run gels. Okay. Was that the first proof of a coronavirus protease?
That was the first proof, yeah. As far as I know, it was the first for any virus that was self-coded. So our model at that point was trypsinogen. Yeah, that cleaves itself.
And so eventually you made a map, a processing map, which you published in 1984 for polio. The authors are Palanch, Olin Q, who was another student of yours, is that right?
He was a postdoc.
Postdoc. I know Olin and Mark Palanch from the CDC. And then Eckhard Wimmer is on the paper, and then you, protein processing map of poliovirus, which I think was the first map of all the ways the proteins were processed, right?
Yeah, now to tell you the truth, I'm foggy. The one that I remember with Wimmer had to do with nomenclature.
Right, that's next.
Okay, well, the fact is I don't remember why Eckhart was on that particular paper. Who were the authors?
So the
again?
first author is Mark Palanch.
Yes.
And we have Olin Q, Bert Semler, Emelianowski, Anderson, Wimmer, and Rueckert. You're the senior author.
Well, okay, Bert Wimmer was involved too.
Yeah,
So
yeah.
sorry, my 87-year-old brain doesn't remember.
All right, so then the nomenclature, 1984, Rueckert and Wimmer, systematic nomenclature of coronavirus proteins. Do you remember?
Oh, yes, I remember that well. You'll like this part.
Okay.
Eckhart had proposed that we needed a nomenclature. And he suggested some of the terminology I can't remember, I guess. 3C Pro or something like that, a certain way of naming it. So at a meeting in Lansing, I heard the people with foot and mouth disease virus, they finally had some sort of a scheme, and they had pretty much identified it, but every laboratory had a different nomenclature. So foot and mouth had one nomenclature, polio had one nomenclature, we had one nomenclature, and actually we should have adopted the polio nomenclature, but at the time, because of the way the proteins moved around in the gels, we couldn't be sure which corresponded to which. And so in order to keep things straight, we gave ours a different name, and the idea was we can rename them when we know what their identities are. So in any case, I knew after the Lansing virus what all of the different group corresponded, which were the corresponding proteins. And so I wrote up a little paper which I showed to Paul Kaysberg, and I said, this would be a way so that all the coronavirus could understand each other, so the foot and mouth people could identify the same protein as the polio people. Paul thought that was a great idea, and then I told Ellie Ehrenfeld about it. She said, you should publish that. Well, the idea was, we had a meeting then, someplace. Urbino, that's where it was. And so I handed this out, this proposal, to everybody. And they said, oh, no, we don't need that. And at the end of the meeting, they all agreed, yes, we needed that. So they had time to think about it, and so we should adopt it. So Ellie said, you should publish that, Roland. And Eckhart says, I'm not going to agree to that if you don't put me on this. So I said, okay, Eckhart, you're an author. And so that's how that paper got published.
So I remember that was my first Euro pick as a faculty member. I'd gone to Columbia in 82, so 84 I went to Urbino. I remember you presenting this, and there was a lot of discussion about it, but you presented it, right? I remember that distinctly, and then yes, you and Eckhart ended up publishing, because he had his own version, I think.
Yeah, he had his own version.
That's why you ended up publishing. So it's called the L434,
Yes.
right? So you had the leader in some picornas, then 4 capsid proteins, 3 in the middle, and then 4.
Yeah.
So
And that all turned out to be pretty good.
it works to this day. We use it, and so we can name the proteins, 1A or 2C or 3D. Works
So
well.
did you change anything?
No, it's not changed at all.
Hey. Been 20 years and they haven't changed it.
It was really well done, really well done. All right, so then, in 1981, someone by the name of Paul Friesen shows up on a Journal of Virology paper, you and Paul, synthesis of black beetle virus proteins in Drosophila cells. So you got interested in insect viruses?
That was our best project ever. Okay, this goes all the way back to classifying picornaviruses. And we have this leftover virus, Rhinovirus, and it didn't grow in anything except mouse brain. And that's not very easy to radio label things in mouse brains. So we were looking for a way to grow the Rhinovirus, and no success. And in Madrid, I ran into John Newman, who was Fred Brown's redhead man. Fred Brown was the head of the World Reference Laboratory for Viruses. So John was a vigorous fellow, and he followed he was one of the authors of the Rhinovirus that said it's a different virus than the picornavirus. But we couldn't be sure the protein wasn't the RNA wasn't being cleaved when they were doing their characterization. That was the problem. Ribonucleases were hard to control in those days. We didn't know that they were on skin. So I'm in Madrid, and John Newman said, well, there's a fellow in New Zealand that has a virus that's growing in Drosophila. It looks like it's a Noda-like virus. And so I said, well, who is that? John Longworth. So I wrote John Longworth a letter. And he said, yes. He said, Paul Scotty has that virus, as black beetle virus, but we're having trouble making it grow. We'll send you the material. So they sent us Drosophila cells, and they sent us black beetle virus. And sure enough, there was virus in there, but it was really a small amount. And we didn't quite know what was going on. We're not good enough virologists to know about persistent infections in those days. So I thought sort of maybe we should try a different strain of Drosophila cells. So we sent for some Drosophila cells, and Paul set it up, and he did an experiment with the ones that we got from New Zealand and the ones that we got from Washington. And then he came into the laboratory one day and have I got it straight, Paul, or is there something in between?
No, that sounds good, Roland.
Okay.
But I knew what was going on all along.
Yeah.
You didn't.
Well, okay. Well, you should have told me. He came in with this huge amount of virus, and we were so elated because we could now apply all these techniques. We could radio label everything. We could isolate the RNA. We could do the gels. And we did it all in about a week. I mean, it was amazing. So the whole world was ours. There was nobody else in the world that worked with this virus, and we had a brand new genus of virus on our hands. So we now knew that, yes, indeed, notaviruses are not picornaviruses. They're a new class of viruses. So we could put it in the textbook there. It's a new class of viruses.
Paul was a PhD student in your lab?
Paul was a student at that time, and he had the whole project to himself. And then later on, we got into flockhouse virus, which was easier. Oh, so one of the basic problems, we didn't have a plaque assay, and it's hard to do virology without infectivity counts. And so Bernie Selling found that he could get plaques with flockhouse virus, the better plaques with flockhouse than with black beetle, and so flockhouse became the adopted virus.
Right. Let me tell you something. Can you imagine that today there are people who think PCR is a good measure of infectivity?
PCR?
Polymerase chain reaction.
Well, does it work? Is it true? Is it
No,
a
it's
good measure of infectivity?
It's not infectivity, it's nucleic acid.
Yeah.
I agree with you. Black assay or some infectivity
Yeah.
measure
Yeah.
is the way to go. It's good to
Okay.
hear. So
Yeah.
Paul told me yesterday you used to take canoe trips together. Is that right?
Well, pretty modest ones. The only trip we took was the what did you call it? We carried the canoe across
Portage.
a portage, paddling portage, right?
Nearly drowned.
And Paul was up front. He was the strong one. We did pretty well, actually. We didn't come in first, but we didn't come in last either. It was fun, but we only did it once. Why did we only do it once?
Well, we did it on the Pelican River too.
Okay, I forgot that. So I have 300 acres with 1.4 miles of river frontage.
Nice.
And it goes all the way up to a 5-lake area that they're imaginatively called First Lake, Second Lake, Third Lake, Fourth Lake, and Fifth Lake. You go through all those and then down the river, and it's about a 4-hour trip. Fantastic. And the otters are playful. They poke their heads up and say, "What are you doing out here on my property?" Yes, that was a good trip.
Cool. So then we move into an era of virus structure, X-ray crystallography. And there's a paper, first paper, crystallization of a common cold virus. That's 1983. And Mike Rossman is on that paper. So you told me this morning a story of how you got interested in crystallography. And it started in Berkeley, right?
Started in Berkeley. You remember Stanley crystallized tobacco mosaic virus.
Right.
Okay, I should say I only saw Stanley about 3 times the whole time I was there, for 3 years in Berkeley. But he came in one day and he said, "Roland, would you like some polio virus?" And well, not really. I was working on an ME virus, but I didn't tell him that. I said, so I had 3 milligrams, more than anybody in the whole world, enough to infect everybody in the whole world of ME virus. That doesn't infect people, but all the mice in the world. So I'm very proud of how much virus I had. I'm a chemist and I'm interested in chemical quantities of virus, not plaques. So 3 milligrams was a huge amount of virus. He said 3 grams, and I didn't believe him. So I asked him again, and he told me 3 grams. It turned out that it was a vaccine virus from Merck. And this had been produced by Maurice Hilleman, who was a very famous virologist. He did the adenovirus work that was causing huge problems in the military. And in fact, while I was at Walter Reed Army Medical Center, we didn't go into this, but Maurice Hilleman was working right next door to biochemistry, so I heard his name a lot. And later he became head of vaccines at the Merck laboratories, and they made poliovirus vaccine, some of which I got in my arm that he made. But it turned out that some of it had SV40 that came from the kidneys that they used to grow the virus, and the vaccine couldn't be used. And so he sent that to Stanley, because Stanley was a virologist, a Nobel Prize winner, and it was logical for Hilleman to think of that, to get rid of his polio virus. So it came in 2 boxes, cardboard boxes that big. And it was all done in safely. It had to be shipped, and so it had to be insulated carefully. It was full of, what do you call, vermiculite. It's full of vermiculite, and then there were paint cans in there, and then inside the paint cans were vermiculite, and inside that was a tube with poliovirus, and each one was opalescent blue with lots of virus in there. So it sat in the cold room for quite a long time. I was trying to think what to do with it, and I decided to compare the proteins. I was doing proteins on ME virus. So I got that out and I put it in the centrifuge, and we separated it with cesium chloride density gradients and concentrates like that. And when I came in the morning to get it out, I pulled out the tube, and there was a white band in there. White, not opalescent, but white. And it turned out it was so thick that it was like a pad of felt. And I took it out with a spatula. I took the band of poliovirus with a spatula. It was so much fire in there. And I dissolved it up, and I did my experiment, and I got a big mess on my urea gradients. And later I realized that this had been treated with formalin because it was vaccine. The proteins were cross-linked, and it was no wonder I got a mess on my gradients. So I didn't have anything useful to do with it at the time. I didn't have any protein synthesizing cell-free system or nothing I could do with it. So I pretty much abandoned that, and I noticed that a few weeks later that the protein had precipitated, and I thought that probably had degraded, and I looked under the microscope, and it was full of beautiful crystals. So Stanley was right across the hall. I went and asked him to take a look, and he said, yep, that's polio crystals. And I said, well, can we do crystallography? And he said, well, no, there's not enough computing power. That was 1965. Well, later then, should I stop here?
Go, keep going.
Keep going. So later, when I came to Wisconsin, at some point, Bob Bach was working on tRNAs, and he had a lot, and he was able to make crystals of TNR. And so they hired Sunderlingam, Mutaya, I think was his first name. He was an Indian fellow, and he was supposed to do the crystallography of sRNA, and I think he did in fact succeed in that. I don't remember much about it. But I went to see him about how's about doing poliovirus. At that time, Steve Harrison had solved the structure of tomato bushy stunt virus, which is a spherical RNA plant virus. So I thought, well, there must be enough computing power now. So I went to see Sunderlingam about it, and he'd been crippled by polio. He walked around everywhere with a cane, and I thought he'd be really enthusiastic to do this. But it was too big a challenge. The technology was too formidable, and he didn't want to tackle it. But there was a student listening to all this, and his name was Jim Hogan. He was a student of Sunderlingam's, and he said, well, I would be interested in it. And so we talked about maybe he could get trained in Steve Harrison's lab. He'd solved the tomato bushy stunt fires. Then he could come back to Wisconsin, and we could get lots of computing if he needed it for crystallography. So to my mind, this was, we're going to get lots of computing. And to Steve is, I'm going to solve the structure of poliovirus. So that sounded pretty good, and he went to Steve's lab. And then Scripps lab was pretty aggressive, and they had a supercomputer out there, and they asked Jim to come out there, and I could hardly say to Jim, no, you can't go to Laurie, you're supposed to come to Wisconsin. I mean, he had all the opportunities, so Jim went to there. But we continued to send the virus. Joe Eisnogel was the guy who made the virus, and we were using vaccine virus because Mahoney was the one that had been crystallized, and that was infectious. So sending the huge amounts of poliovirus through the mail, well, that was pretty iffy, and I thought there's going to be lots of technical problems trying to get that done. So we did the vaccine. And then Jim got an offer from Harvard, and that really complicated things. So I told Jim that I thought if he didn't want to work with the vaccine virus anymore because it was not stable in the beam. He wanted to work with Mahoney, and I didn't want to do that. So I suggested he work with somebody where he was. That was a person named Marie Chow that worked with David Waltapore at one point, and she had her own laboratory by now. And so they did do that, and I don't know much about it. I wasn't involved in it. But meanwhile, Rossman and I had a conversation about it, and he said he was going to do poliovirus. And I think Jim was still at Scripps at the time, although I can't remember. That's right, yeah. And I thought Jim was going to come to Wisconsin, and I didn't want Rossman working on poliovirus. So I said, gee, Jim is working on that, and he's just a young guy in his career, so why don't you work on rhinovirus? No, he said, we got the sequence for poliovirus, and he said, we know about the stability, we don't know anything about rhinovirus, we don't have the sequence for rhinovirus. And well, we talked about it for quite a while, and I told him that I'd give him support, and it'd be good if you weren't both working on the same problem, then you've each got a result. Well, he finally decided, Jim had a career, I'll do the rhinovirus. And I have to say, I tremendously admired Rossman for making the decision that way. So that's how it all got started. And he was way behind because there was no sequence. We didn't know whether you could get a stable crystal out of rhinovirus or not. And so we didn't know if it's going to be stable in the electron beam. So we went ahead with it. It was 1981, and I see by 1985 we had the structure. So I'm sort of impressed how fast that whole thing was. It didn't seem fast at the time. But we had quite a few meetings here. We called them the whisper meetings, and we enjoyed doing canoe trips, and sometimes we had trouble holding the people together because the Wisconsinites they had their own projects, and they would sort of abandon ship when the meeting was half over, and the people from Purdue weren't too happy about that, and I wasn't quite sure how to hold everything together. So we had our problems, but things worked. And Barbara Sherry started making neutralization-resistant mutants, and actually Ann Mosier was making neutralization polio mutants. So we were working on both things, trying to keep things every together. But things dissolved with Jim's lab. Jim was secretive and he didn't communicate much, and we pretty much lost touch with Jim. But of course, things were really humming with the Rossman laboratory, and well, Rossman was using a new method. So Jim was using a heavy atom replacement method, but Rossman was using what he called the molecular replacement technique, which was very controversial. So nobody believed it except Rossman. And basically the idea was he had found when he was working with Proots that the hemoglobin fold for the alpha subunit and the beta subunit were both similar to the myosin subunit. In fact, there were a lot of similarities with the alpha and the beta sheets and so on. And so he thought, well, what if we assumed that there was similarity with tomato bushy stunt and with rhinovirus, and we simply put in the basic structure of tomato bushy stunt and then work from there, and damned if that didn't work. So I think that's how Rossman won the battle. But there was one last thing. I think somehow Jim must have known how well Rossman was doing. I don't know, maybe Rossman told him. But the stability in the beam was one of the decisive things. When you put that X-ray beam on, it destroys, it's very hot, and it destroys the structure that you're bouncing your waves off. And so you have to get your diffraction pattern before the crystal disintegrates. And I'm not sure exactly why, but the synchrotron beam in Chicago had the right properties that he could get a lot of data before the crystal dissolved. And so he was able to go to Chicago to get that done. And so was Jim Hogan, but they would not permit Jim in there because he was working with Mahoney. So Rossman got his data faster because there was no safety problem with the rhinovirus. And that is why he won the race. It was the safety issue is what it came down to. So it's interesting how things turn out when you do it. And also I should say, Rich Colano got the structure for rhinovirus 14 and was willing to give it up. That was also key. Without that, it wouldn't have been possible to get the structure.
I remember that at a subsequent Europic in France, and you were there, both Hogan and Rossman presenting their structures.
Yes.
And they were quite
Yes.
fighting each
And
other.
it really was unnecessary. It was an ego business about I was first. That's what it was all about. And Hogan was really irritated because he wasn't first, and I'm sure Rossman would have been really irritated if he hadn't been first. That was trivial. I mean, they both had magnificent structures.
So then there's a series of papers where you look at these antigenic sites. As you mentioned, you selected monoclonal, resistant. And I remember going to meeting after meeting, and you would give talks, and you were collaborating with Rossman. You were so excited about this, being able to look at these mutants and then look at the structure and see where the epitope was, right?
Well, okay. So that sounds sort of really not very interesting, but the fact that it was riveting. And the reason was when Rossman had the structure, this all happened in one day. The data came in sheets, so that there were segments of data where they could look at the electron density like that. And they piled up 50 of these sheets. Somehow they got an image of the electron density on each sheet, and they piled them up. They had 50 of them like that. And you could just see all this electron density on the sheet, and you could take it off and follow it down, take it off and follow it down. So you could actually see side chains like that were sticking up or winding over and so on. You could trace the backbone, and you could see the side chains coming off the backbone. But it was pretty stressful. I mean, you had to look pretty carefully to work that all out. And of course, we had the sequence. This should be arginine, and this should be aspartic acid, and this should be glutamine, and so on. So we knew exactly where in the sequence which amino acids were substituted. And so we could look, follow the chain up, and when it came to one that was exchanging, that would give you an escape mutation, there would be the side chain sticking straight up right out of the surface of the virus. I mean, the things were all over, and all of a sudden this side chain, the one that substituted, is sticking straight up. And then you keep going around, you come to the next one, and there it was, sticking straight up. And without exception, there must have been 20 mutations. All those amino acids were exactly pointing up where the mutation occurred. You could just see how the antibody had to be touching it. It was thrilling. And whoever ever saw in atomic dimensions what they were working on, it just didn't happen before. So we absolutely knew there couldn't be a mistake, and you never have that feeling in research. There's always a feeling something's wrong.
I remember those days because I met you at meetings, and you were so excited about this, as you are now still.
It was exciting.
And I wanted to mention some of the names because we've mentioned them because it's quite impressive, the people you've trained. Barbara Sherry was involved in that. And she's, of course, on her own for quite some time now. And who else? There was another person. Oh, so now just a few more papers and we'll wrap this part up. 1994, there is a Flockhouse paper, reconstitution of Flockhouse provirions with Annette Schneemann and Tom Gallagher. So they were both in your lab as well.
Yeah.
And they went on to be scientists on their own. So quite impressive.
Okay, I took it for granted, but thank you.
And then there's another interesting paper in 1996 with Paul Alquist, complete replication of an animal virus in yeast. Remember that?
Well, yeah. That is an absolutely amazing virus. It grows in everything you put it in. It grows in tobacco, it grows in yeast. Who else does that?
No, it's a great system for working on host factors as well. And your last paper seems to be 1998, where you start looking at virus antibody complexes by cryo-electron microscopy.
Okay, that's not me, that's somebody else.
Well, you're on the paper.
Well, okay. Well, what's the name? What's the subject of it again?
Antibody-mediated neutralization of Rhino-14 explored by means of cryo-EM and crystallography of virus.
Oh, yes. Okay, now that was a Tim Baker paper, right? Mm-hmm. Oh, well, okay, I remember that. Yeah.
And I think it's interesting because it's probably the beginning of cryo-EM, and now it's, of course, huge.
Yeah.
In
Tim
virology.
Baker was the big man there. he did absolutely fantastic work. It was breathtaking, all the structures that came out of Tim Baker. Now 20 years have gone past, and I know nothing about it. I don't know what happened to Tim Baker. And I see that cryo-EM has now progressed to the point where you can actually do it on not completely pure things anymore. So that's going to replace X-ray crystallography, I guess.
Can go down to very low resolution.
So, boy,
Yeah, it's
I'm really
amazing.
envious, I have to tell you. I sure wish I could do that.
I mean, a lot of it I understand is the electron microscope, but also the computational algorithms have really made a big difference. It's remarkable. So then let's talk about the life after science a little bit. You retired in 1996, right?
December 31st, yes.
And so, I mean, many people would say that you probably had another 20 years of science in you. Michael
Yes.
Rossman has kept going.
Yes. Well,
Why
I
did you
did.
retire?
I did, but I was exhausted. And the way I knew was when Friday came, I had a research meeting, and I couldn't wait to get out to go up to Rhinelander. And that's wrong. I mean, that's not the way I should have felt about my research. So I just felt exhilarated when I got up there. It was a whole different life. Up here, there was always a meeting. I had to worry about a paper. There were letters of recommendation to be written and a grant, and worrying about people's salaries and the laboratory, teaching that laboratory. So it just felt so good to be relieved of all that. I think I was burned out is probably the correct thing. And actually, I never looked back. I learned forestry. I reinvented myself, I think, is the right thing. And I felt very good about reinventing myself. So I liked learning about forestry. I took courses in botany, and I acquired a huge respect for botanists. I sat through the lectures, and I had a lot of trouble understanding it all, but I realized that it was really a course in evolution. I mean, you start from the simplest photosynthetic organisms, and you get into sex, and all of the different things that plants do, and it all came first with plants. I mean, everything that's there in plants shows up in animals later on. And I was in awe of botany. I couldn't possibly absorb all that, but I certainly understood what a treasure botany is, and how much knowledge has come from botany that we don't even pay attention to.
So you spent most of your time there, north?
No, I spent half my time in Rhinelander and half my time in Madison.
Okay. And
And that was a problem because I tried to take coursework. You really can't make it work by skipping half the lecture. So that was an unfortunate realization, but I wasn't determined enough that I was going to miss my Rhinelander time. So.
when you were up there, what did you do?
Well, first, I had an aging father, and that did require some time. So more and more time as time went on, he eventually had to go to a nursing home. Things got to a crisis nature. So that was actually quite a bit of time consuming. But I learned the foresters. I learned how to identify plants. I planted trees, and I had one really disappointing thing. I bought 200 blister rust resistant white pines. Blister rust is a pretty widespread disease of white pines. I wanted to have white pine forest. That was the original forest that was up in Rhinelander, and the ones with the 250-year-old trees. And I wondered why they didn't plant white pines. I was going to do that, and I learned later there's a very good reason they don't plant white pines, and that's because they have disease problems. So they have a terminal weevil, tip weevil, that eats at the terminal buds, and it deforms the trees, and that causes problems. And then you have blister rust, and then you have deer foraging. They eat the young trees and they deform the trees, so you can't get good timber. So it's hard to get good timber out of white pines, and all of these problems came from Holland. I mean, they were not native problems. So they were diseases that moved into the country. Now it's red pine. Well, okay, there's a whole bunch of things like this that are involved in forestry. Deer foraging and and different kinds of rabbit damage and and so on. That you learn about. I learned about it, a larch sawfly that didn't involve me anymore, but we have no larch on the land. And the reason is because the sawfly came through in 1914, swept the entire country and killed all the tamarack. It was like the chestnut blight that got wiped out that way. Okay, lots of stories like that.
So you like spending time alone in the woods?
Oh, yes, but I have to say the television has encroached more and more in my life. So first of all, I find now I'm watching a lot of politics, which I shouldn't do. But I also discovered the great courses. And the first great course I took was in geology. At first, I thought I know all that. Then I started to explain it to my wife, and I realized, no, you didn't learn all that. So I go back and learn it again, and then I took a course in astronomy from a Berkeley professor, Filipenko, Alex Filipenko is a young professor. 96 lectures, and that was really exhaustive. It was started with the origin, the Big Bang, and then the formation of elements from hydrogen, and that was thrilled me because I was a chemist, and I learned the periodic chart and the rules for the s orbitals and the p orbitals and all that. It never occurred to me, how do we know all that? Well, so the origin of the elements was of great interest to me, and I still don't understand it all, but I know there's the proton-proton chain, and there's the alpha, the triple alpha process, and there's the slow and fast neutron methods in the supernovas. So learning those things is good, but I forgot most of what I learned. But I found it thrilling. All the knowledge that was developed while I was busy being a virologist was coming out. Tectonic plates and all that, that was all new to me. And I'm still learning that. So I have those courses at home and I tune into them. So that's what I do when I'm alone.
So I understand you live on Rueckert Road.
My father was a pioneer. He bought 200 acres in the wilderness, and they built a road back there, and so they called it Rueckert Road.
That's pretty cool.
So it was my father's
nice
legacy.
So besides everything I've said today about your career, one thing I do remember, you have very clear writing in your science papers, and I went and I found online a little description of what you do, and I just want to read a few sentences here because it's lovely. When the trails are clear of snow, much of my time is occupied by keeping the roads clear of fallen timber, my source of firewood. I patrol my 3 miles of logging trails on a sturdy ATV, carrying necessary tools, chainsaw, pole saw, pruning saw, lopping shears, and pruning shears as the case may be. Bucking the trees into segments, splitting them and piling the firewood is invigorating in the dry, cool fall, but sweaty and disagreeable during the humid mosquito season. I also work on aesthetics, keeping brush from taking over on trails, but the heavy lifting of forest management is done not by me, but by a logger. And there's more, I'll put a link to that, but that's lovely.
Somebody read that.
Of course.
I didn't write it in vain.
Nope.
Well, I glow when you read it. I feel good about that.
It's wonderful. It's your writing. I recognize it totally. So you also on that website, you were named the Oneida County Tree Farmer of the Year in 2001.
Yeah.
That's pretty cool. But you don't keep in touch with virology anymore, right?
No. So I get ASBM B,
Yep.
and I read that, but there's practically no virology in it. And I do go to the virology meetings, but most of it I have trouble that the things go too fast, and the techniques are new, and I'm not familiar with them. I can't follow it. And technology used to fascinate me, but now I don't care anymore. I can't use it. What's the point in learning it? So I've just fallen behind.
So the last time I saw you was I think last year at ASV. It was held here in Madison. I don't remember, I sat with you and Sid Grossberg.
Yes, I remember.
For a while, and that's when I had the idea to have a little conversation with you. And let me end it by just asking a couple of things, more general question. What do you think is your most important contribution as a scientist?
Well, I'm proudest of most of my students. I mean, science is about human beings. And science is a magical thing. I think it changes people. My religion is science. I probably horrify some people, but I do believe it is the closest thing we have to truth.
So I think I know the answer to this question, but let me ask anyway. What would you have done if you had not been a scientist?
I can't imagine. The nuns wanted me to be a priest. I didn't think that was a good idea. I might have been a good one, actually.
Yeah? Have you read any good books lately?
Well, I just told you about Steven Weinberg's book, To Explain the World, and it comes right back to what I said about science. He describes in that book the earliest glimmerings of fragments of science, the earliest part of which was mathematics, learning mathematical relationships, and that you could relate nature to certain mathematical things. But he progresses how the loss of knowledge and the fights that people had, and in fact, executions for beliefs. So you know about some of those things with, what's his name, the one that was burned for his, saying this, the earth goes around the sun. But anyway, I came to appreciate what a precious thing science really is and how fragile, easily lost it would be because of all the special knowledge that we have and how it's divided up between people. So the university to me is an increasingly precious place. And the people who grow from it are precious people.
The guy you're thinking of is Giordano Bruno. He was
Bruno,
burned.
that's the name I had on my tip of my tongue.
He was burned at the stake for saying the Earth went around the sun. All right, one last question. RNA or DNA?
I don't know. RNA came first, I'll say that.
Okay, that's good. All right, that's TWiV 529. You can find it at microbe.tv/TWiV. Most of you listen on your phone or mobile device. You use an app. Please search for TWiV, subscribe so you get every episode. It helps us to see how many subscribers we have, so please subscribe. Send your questions and comments to twiv@microbe.tv. And if you like what we do, consider supporting us. You can go to microbe.tv/contribute for the different ways that you can do that. My guest today has been Roland Rueckert, Emeritus Professor at the University of Wisconsin-Madison. Roland, we didn't talk about this box of tissues here. I just want to show our viewers. It's a box of tissues called AVERT. Virucidal tissues, kills major cold viruses. Has citric acid, malic acid, and SDS in it. You involved in the development of these?
Of course, when the Rhinovirus thing came out, we got a visit from scientists at Kimberly-Clark. They make these, you recognize what's in here, and they wanted to make something that would kill cold viruses. So they came and asked us if we'd do some measurements for them. Their idea was to put SDS and acid in the Kleenex, and how did it work? So they hired us to do some tests, and what we did was just put a drop of virus on a piece of tissue, and then drop the tissue into let's see, we put the virus on the tissue and drop the virus into stop the killing. So neutralize it. That's what I'm searching for. We neutralize it after we put the virus onto the tissue and see how much virus was there, and then we'd make a plot. And the half-life of the rhinovirus was 9/10 of a second on the tissue, so it worked pretty fast. But when they had to charge a pretty high price, I think it was like $5, something like that, in order to make money when they sell it. Marketers did all that, and they actually put it on the market. I saw it in Rhinelander in one of the stores. But apparently, people didn't buy it, and probably one of the reasons is that it burns. It's got citric acid in it, and if you snuff up backwards, the citric acid goes in your nose and it stings. And maybe that was a reason people didn't buy it, so it went off the market again.
There's a newer product now you can get from Kleenex that.
Oh, I didn't know that.
Yeah, it's and they say antiviral on it, and
Well, who makes Kleenex? Is that Kimberly-Clark?
I think so. Yeah.
Well, then it's probably a similar product.
It's actually let's look antiviral Kleenex. I got a call from a reporter once. Yeah, Kleenex antiviral face tissue, $1.59,
Oh.
much cheaper.
A lot cheaper.
And they put the virus on it, and they do a plaque assay to measure infectivity. It works. Yeah. Roland, thank you so much.
You're
talking.
welcome. Thank you for all your research.
My pleasure. I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASM for their support of TWiV, Ronald Jenkins for his music, and for the local PhD students for inviting me here in the first place. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.